Hello, and welcome to our poster about the bacteriophages of Rhodobacter capsulatus and its gene transfer agent, RCGTA. Rhodobacter capsulatus is a purple non-sulfur photosynthetic bacterium in the alpha Rhodobacteria that has been used to study photosynthesis and pigment biosynthesis. RCGTA is a phage-like particle known as gene transfer agent that allows for the transfer of genetic information between host strains of Rhodobacter capsulatus. Remarkably, its genome is present throughout the genome of Rhodobacter in 3 or 4 different islands that are much larger than the amount of DNA that can be packaged by the RCGTA particles. Hence, it is essentially a tool for lateral gene transfer, or in the hands of a geneticist, a way to make transduction happen. The Rhodobacter bacteriophages that have been sequenced thus far are a total of 26. The genome sizes range from 36,000 to about 102,000, excluding Zuper that Rigalvi will talk about at this meeting. We see a wide variety of chromosome organization, including things that we're probably used to with the mycophages, including circularly permeated genomes or defined cohesive ends. But we also see mu-type phage and a P1 type of packaging. Our phages can be organized into 6 clusters and 3 singletons. The clusters are clearly visible or organized in part by nucleotide sequences seen as in this Shepherd plot. So you can see that the clusters are readily observed as areas of shared sequence in this Shepherd plot. Based upon the clustering, we also have done TEM images, and you can see examples of each of these clusters, as well as 2 of the 3 singletons, Simona Hostad from Sweden and singleton RC Zahn. These are all in the Cyphoviridae. Something that's probably very familiar to everyone in this group is a Famerator map, and you can see based upon a quick view of this that the organization of the genomes of these Rhodobacter phages is fairly similar to what we see with the other with the phages in the mycobacteriophage groups. We also did a splits tree to try and analyze the relationships based upon shared genes. And again, we see the same clusters fall out, and the singletons, Zahn and Simona Hostad, stand out as very different from everything else. These other 2 singletons shown here are actually singletons that were sequenced by other people. One of the questions that we wanted to address when we started working with Rhodobacter capsulatus phages was whether they shared any similarity with GTA. Because RCGTA is a well-known gene transfer agent in Rhodobacter and causes essentially a phage-like infection, we wondered and thought that maybe there'd be shared gene content. This is true for some of the clusters and some of the singletons, and notably, there are 4 proteins that are shared amongst many of these. These proteins are involved in host recognition, including the distal tail protein, a protein now known as the hub protein, and this protein, which has been annotated as a GTA Timbarrow-like domain, is now known as Megatron. And finally, there's a peptidase or cell wall hydrolase here that can cause cell lysis to occur. I'd like to thank my collaborators, including, of course, Dr. Rick Alvey, who will be talking at this meeting, and student members of our laboratories, student members of the C Lab, and collaborators at other institutions, including Dan Russell, Becky Carlina, and Steve Crisson. Thank you for your time.